Tried to judge the attendance for these meetings is always so wonky. We're supposed to go by half of who RSVP'd, but for this event we had 90 people RSVP'd.
Wow.
Yeah, I think we'll start now. Aaron has a lot of information to present, so we're on a bit of a time budget here today, and so take notes. This is going to be a very informational meeting, and glad everyone can make it. See a lot of new faces, and so if this is your first time at a Portland Psychedelic Society meeting, welcome. Just gonna have to go over a few things. There are no services allowed to be exchanged or solicited or any type of substances within these meetings. This is purely educational. Okay. Now, raise your hand if you are a police officer. Yeah. So I mean, it's purely educational, and really glad that everyone's here and be able to witness things like this happening in 2019. It's a cool time to be alive. So, glad to do. Yeah, and also, the donations, we have this over here. If you have to leave at any time throughout the meeting, we would really appreciate if you could donate $5, $10, whatever you think this meeting is worth it. It does cost money to rent out this business room. And then at the very end, when we're closing out, we'll pass around a cup, and that's about all I got. And so without further ado, everyone, Aaron, please.
Well, hello everybody. Thank you so much for taking the time out of your days, and I just want to be here to thank the Sage Wellness Center and to thank all of the researchers who have contributed to this research, all of the spiritual leaders, everybody who's taught me everything I know and given me these amazing opportunities, including the Portland Psychedelic Society. So thank you all. So this is also a presentation by TRIPS, which is a nonprofit organization I'm in the process of starting, which is translational research in psychedelic studies. The goal of TRIPS is to essentially provide education and one-day research for psychedelics, and to kind of hopefully advocate for policy change. And I believe education and research is one of the best ways to have that be done. Here's a little background on who I am. I'm a graduate student at the National University of Natural Medicine. I'm going for a Master's of Science in Integrative Medical Research, which is both conventional and naturopathic medicine. And kind of the goal in that is to establish more research on natural medicine, because there's a lot of benefits that it can provide us, and kind of one of the caveats about that is that there's not enough research done, so we really don't know what works or what doesn't work. So this degree is fairly new, and I plan to be a part of it to help change that field. I'm also a research assistant at Oregon Health and Science University in 5 different labs: the School of Nursing, Developmental Brain Imaging Lab, ADHD Research Study, the CDC National Spina Bifida Registry, and the Portland Alcohol Research Center is having a study up there as well. I'm a research council member for Northwest Noggin, which is a neuroscience outreach group. We go to K-12 public schools and so many more. We build what I have here and kind of dangling around the room are these pipe cleaner neurons. So we like to kind of go to kids and teach them about the brain through art, because it's a very easy way for them to get a hands-on experience and kind of be able to understand that. So there's all kinds of different neurons that I have around here, and I'll be using them slightly to help as visual aids throughout the presentation. So, I'm also, as I mentioned, the director of TRIPS, and I'm a student member for the Society for Neuroscience, which is the largest neuroscience organization in the world that is working towards advocate policy and kind of collaborate and bring researchers together. There's going to be their annual meeting in Chicago where I will be presenting a poster. And I'm also an honorary member of Portland State's Neuroscience Club. We currently do not have a neuroscience major there. All of our classes are kind of pushed into the psychology department, but we're advocating for a neuroscience minor that will actually be jointly with OHSU. So there's my contact information up there. I also have my cards over there as well. Feel free to take any pictures you want throughout this presentation. I'm also happy to send you these slides if you take down my contact information, and answer any questions that kind of go further on the topics that we discussed today. Or if you have your own event and you're interested to kind of bring me down, I'd be happy to present free of cost. And because we have so much information to cover, it would be greatly appreciated if we could hold questions until the end of the presentation. If you have a question that's really important, kind of write it down and then we can discuss it in the Q&A after. However, if I fail to describe a terminology or I didn't do it that well and you're a little confused, feel free to kind of raise your hand up, be like, oh, can you kind of describe that a little bit more? I totally would be happy to go in there. We're going to cover some pretty big neuroscience topics that even went into my advanced neurophysiology class. And there will be some level of generalization applied here because I don't have time to go fully in depth on a full neuroscience lecture, so there will be some level of generalization applied to this presentation. And disclaimer, I'm going to get my disclaimer voice ready. The views and presentations expressed in this presentation are those of the author and do not necessarily reflect official policy or position of OHSU, PSU, NUNM, Northwest Noggin, the Society for Neuroscience, Neuromeditation LLC, the Imperial College of London, MAPS, and MAPS Canada. So we're going to go down the rabbit hole a bit today and kind of discuss how psychedelics function. And we're going to focus on specific research that is being currently conducted. I have all of these papers stored, and I'd be happy to send them to you if you would like to read these scientific papers a little bit further. There's a lot of paywalls that are kind of around research like this, which I'm very much against because you've already paid for this research with your taxpayer money, so why have to pay for it a second time? And I also want to make a shout out to the first neuroscientists, who were the ancient Egyptians. They believed the brain was cranial stuffing, which might not be entirely accurate, but they also provided the first written account of the organ and described its anatomy, including cerebral spine fluid. This is Egyptian for brain. So, your brain is made of cells, neurons, kind of like what I have all around here and all kind of dangling around there. And these neurons carry electrical messages. And they connect chemically across synapses. So the neurons don't actually touch one another. You might have, so this is kind of the tree-like areas called dendrites, which means actually branches, like tree branches, because they look quite similar to that. And then this would be the wire part, is called the axon, where messages kind of flow down and they travel out through the axon terminals, kind of like a terminal at a bus station or at an airport. And so your neurons are kind of wired up close to one another. So here would be an axon terminal and here would be a dendrite. So they don't actually touch. There's a small little gap between the two of them, and that's called a synapse, which we kind of see here. And then they actually communicate through both electrical messages, and then those electrical messages and currents will release neurotransmitters, which then go but in the synapse, in that little gap, and they will touch receptors on the dendrite, and they don't actually enter the other dendrite, and they cause another message to kind of go. And they pretty much say one of two things, either inhibitory, let's turn it off, or excitatory, let's turn it on. And that will kind of cause a whole cascade of different reactions to occur. So all hallucinogens act at one type of serotonin receptor called the 5-HT2AR. And just to break that down, 5-HT, serotonin. 2A refers to a postsynaptic receptor. So again, we have the axon terminal, and we have a dendrite. So that little gap right there would be the synapse. So the 2A receptor is a postsynaptic receptor, so it's actually on the dendrite. So these would be the 2A receptors, and R simply stands for receptor. And so this is basically how they communicate to one another, and there is 15 different serotonin receptors. So the 5-HT2A receptor is one of 15 different serotonin receptors. And here we kind of have a graph. This would be the potency of the hallucinogenic drug, and this would be how well it binds to this receptor. And yeah, it's a little hard to see, but again, I'll be happy to send you these slides. And kind of down here, we have LSD, which is very, very potent because it binds to the receptor so well. And we actually have DMT up here, which at first I thought that was a little interesting because a DMT trip tends to be a little bit more intensive an experience than a LSD trip. However, if you think about the amount you're consuming about 100 milligrams of DMT versus LSD, you're consuming micrograms, about 125 micrograms. So it's much smaller doses. If you consumed 100 milligrams of LSD, that would be quite an experience, possibly even fatal. So hallucinogens affect the gating of sensory input. You have a part of the brain that's called the thalamus, which is kind of that little island-shaped thing. So here's the brainstem coming up from down there, and the thalamus sits on top of the brainstem. And you can kind of see why it would be important for sensory gating. You can kind of think of it as the old operators of like telephone wires that would pull wires out and plug other wires in. So that kind of sends messages from your brainstem to other regions of your brain where they're supposed to do. So hallucinogens actually shut this part of the brain down, which is very interesting. So you can kind of see the thalamus here, and then all these different messages kind of travel out throughout the rest of the brain. This is courtesy of the Allen Institute of Brain Science. Got a little off topic. So here we can see a normal brain. It's a little hard to see, but there's connections that occur naturally, and the thalamus is again kind of sending those messages to where they should be. So when the thalamus is shut down, you actually have this flurry of messages that kind of connect to different regions. And the different colored regions are different lobes of the brain. So you can see there's a much greater connectivity that occurs. And that kind of has to do with synesthesia, why you're able to perceive, like visually see things like sound under the influence of that. It's because these connections are now from your auditory cortex are now connecting to your visual cortex, and it all kind of like mixes together. And that's also why when people consume a psychedelic, it's not always the same experience. For example, other substances like opioids, cocaine, methamphetamine, they all kind of act on one specific thing and they kind of do the same thing, versus psychedelics cause this to be shut down, and all these connections occurring can cause a whole wide range of effects, which is why you can take a psychedelic 1 week, have an experience, take it 2 weeks later and have a completely different experience. They're very much related to the surroundings of your environment. There's also another important part of the brain called the locus ceruleus, and it's the noepinephrine network, which also is related to your kind of adrenaline network. It's your orientation. So it's what orients you to different ways. So if you were to see a spider on the roof, you'd be oriented towards that. Or if you were to walk out into the road and see a car coming at you, your brain would orient you towards that. So you could make the decision, oh, I need to get out of the way. So psychedelics tend to hijack this network and orient you to things you might not normally be oriented towards, like this chair or your hand. And where are these receptors? They're everywhere. So the cortex, which again is this kind of outer wrinkly area of the brain, the olfactory cortex, which is smell, the hippocampus, which is memory, the basal ganglia, which is quite complicated. It's attached to many structures, although it is responsible for a good portion of the structures in your limbic system, which is your motivation network. And just because it's connected to that network does not necessarily mean that it turns on motivation to these substances, as we'll see a little later. The thalamus, as I mentioned, kind of the gating area. Cerebellum, which is actually your movement, so balance and being able to move. So I always find this interesting. 60% of the neurons in your entire brain are in this little area right down here, which is the cerebellum. And that's because it takes a lot more neurons to coordinate movement rather than to be like, I want to eat a pizza right now. And then, of course, we have the brainstem and the spinal cord, which how messages kind of travel throughout the nervous system into your brain. And then another very interesting part is the default mode network. So that's kind of your rumination network. It's the network that allows you to kind of think, what do I want to eat tonight for dinner? Or what do I want to do on my day off next Tuesday? So this is really critical for self-reflection, awareness. So we actually find that psychedelics, under the influence of psychedelics, shut down this network, which is why it might be a little hard to plan out something while you're in the middle of an LSD trip. And this kind of promotes mindfulness. An interesting thing is actually there's other ways to shut down the default mode network other than taking psychedelics, such as meditation, which kind of promotes this mindfulness and the ability to kind of look inwards on yourself. So I'm just going to go over the types of psychedelics. We have the classical, the empathogens, entactogens, the dissociatives, and then down here are my not so favorite ones, the deliriants, and then the research chemicals. So again, there's a trigger warning. I'm going to be showing some substances on the screen, so if that bothers you, this might be a good time to step out for a minute or so, but this might not have been the best lecture to come to if that is something that bothers you. So we have the classical psychedelics, LSD, so this is some LSD blotter art down here. We have psilocybin cubensis, which are kind of these mushrooms up here. We have DMT, so these would be some pure DMT crystals. Mescaline, so this is pure powder that has been extracted from mescaline. And then we also have ibogaine up there. Then we have they all act on the 5-HT2A receptor. So then in pathogens and entactogens, they also act on the 5-HT2A receptor, but they do a little bit more than that. They are reuptake inhibitors, so they basically prevent certain neurotransmitters from being pulled back into the neuron. You have more in that gap, the synaptic cleft, allowing them to bind more to receptors and kind of getting a similar action. For example, cocaine is a reuptake inhibitor for dopamine. So it blocks dopamine from getting sucked back into the neuron. You have more dopamine in the cleft binding to the receptors down there, which causes that kind of rush of euphoria. So MDMA, MDA, MDE, MDAI, MPDP, MDPV, all the word salad ones. So they're reuptake inhibitors for serotonin, so they actually are SSRIs, which is interesting, and norepinephrine, so they're SNRIs, and weekly dopamine. And they also release oxytocin, which is possibly why they're associated with more of the loving chemicals or this kind of like connection with other people. Then we have the dissociatives, so that would be PCP, ketamine, salvia, and DXM, which is common cold medicine that is available over the counter. It's in NyQuil, it's in corticotropin cough and cold, Robitussin, and these are all NMDA receptor antagonists. So it can be either an agonist, so it binds to a receptor, turns it on, which is what psychedelics do to the 5-HT2A, or an antagonist blocks that. So NMDA receptor, really important in learning and memory. There's this really interesting experiment called the Doogie mouse, where they actually find that these receptors are open for longer periods of time when you are younger, which promotes learning, which is why it might be a little easier to learn when you're younger. So what they did was they had an older mouse, and they took the younger mouse's receptors, put it in the older mouse, and the mouse was able to navigate through mazes better, suggesting that these do play a pretty important role in learning and memory, so it gets shut down under the influence of a dissociative. And they actually act on the opioid receptor, a specific one, which is very interesting. But these kind of produce an effect like you're not in your environment or you're kind of pulled away. They actually did this really interesting experiment where you don't have pain receptors in your brain, so they can actually be working on your brain while you are conscious and awake. And they found this one network, and they stimulated it with electricity, and the person described feeling like they were being lifted out of their body when that network was stimulated. They took away the current, the person got sucked right back in. So it's very much associated with where you are in your space and time. Then we have the deliriants, which are nutmeg, Benadryl, yes, Benadryl is a type of hallucinogen, Dramamine, deadly nightshade, and Datura. These act on, they're anticholinergic, so you have the acetylcholine receptors, which are at the neuromuscular junction, so they kind of affect areas where your brain is able to communicate with your muscles, including your heart, which is why these can be deadly. Oddly enough, they are all legal. And then we have the research chemicals, so all the word salad ones, the DOX series, the NB series, which we're going to talk about a little later, the 2C series. So these are all probably the most dangerous class of substances. There's definitely a few golden ones in there, but they have been invented very recently. For example, people have been consuming mushrooms for thousands of years. We know the interactions, we know the long-term effects, That is something we don't know with these substances. So that's something to be mindful of and careful of, especially if you're consuming a substance, to make sure that it is what you think it is, and not one of these research chemicals. This is actually 2,5-I down here, and you probably can't read it, but it says plant food. So I don't know what kind of plants you're growing if you're giving them 2,5-I. And then Alexander Shulgin and Anna Shulgin were kind of some of the people who really pioneered this field, and this is some of the books they wrote about kind of their experiences and how to synthesize these chemicals. So again, let's go back to the 5-HT2A receptor. So I mentioned it's one of 15 different receptors for serotonin. It's the main excitatory one, so it turns things on. And there's a GPCR. There's two main types of receptors. You can have one. Now, remember, the neurotransmitters don't actually enter these receptors. They touch them, and then they cause them to either turn on or turn off. So an ionotropic receptor basically is closed. A drug will bind to it. It opens up and allows sodium ions or other ions to pass through. Sodium has a positive charge. Electrical current is generated. These are a little different. So these have a little G protein attached to the bottom. When a drug binds to it, the G protein is released and it goes inside the cell and causes intercellular changes. So the ionotropic ones tend to be the quicker, like instant response, you feel it, versus the GPCRs can cause longer term effects, which is what we're going to see under psychedelics, which is really interesting. Although they're excitatory, they're also a little inhibitory in certain areas, such as the frontal cortex and the visual cortex. And your brain does not like it if there's gaps in information. So it'll actually look to different areas that are next to it to pull that information from, which is actually how we experience hallucinations. It's based on, like, our brain is like, okay, well, this part's missing. Let's fill in the gap based on what's around me. So that's kind of how we have these hallucinogenic kind of visual experiences. There's also a really interesting thing about that with phantom limbs. Sometimes people who lose their arms are able to kind of perceive that they still have their arm, and that's because the brain is looking to regions right next to it and picking up. There's a really interesting experiment that was done with a Q-tip. So you don't need a lot of money always to do neuroscience. And the person got a Q-tip, and they tickled a person on their cheek who had a phantom limb, and they were like, wow, that's my finger, that's my little pinky. And they tickled over there, they're like, oh, that's my thumb. So it turns out their connection to their face kind of connected to other regions that they perceive their hand. So every time they moved, which is really common to move your mouth, they perceived their hand was there. So they're in high levels in the cortex, which is very important for learning, memory retrieval. They're also concentrated in the posterior cingulate cortex, PCC, which is a central mode in the default mode network, which is kind of what I was talking about a few minutes ago. But they're not that common in the motor cortex, which may be why psychedelics had such a strong visual and cognitive experience, but you're not so emotionally incapacitated. You can still walk, you can still move, you can still do what you want to do. And then their agonism, so remember, antagonists turn things off. So they're agonists, so they turn on naturally. They're not just there for psychedelics, they're there for a natural reason, and they seem to be there for the stress response. And this finds to enhance cognitive flexibility, improve associative learning, and they actually can grow new neurons in the hippocampus, which is responsible for memory, which might be helpful for kind of learning new things. And it's kind of good to have an element of flexibility if you're going through a stressful situation, so you can kind of adapt to that stressful situation. And then here's 15 different serotonin receptors. That one would be the 5-HT2A. So let's talk a little bit about psychological. When you kind of experience that, it's more well-being, optimism, openness, ego dissolution. And that's really helpful when there's a need for flexibility or when rigid thought patterns kind of need to be broken. I like to kind of say that psychedelics make you question everything you might know might not possibly be correct. So it kind of gives that room to consider other perspectives and other things, which maybe is why you see a little bit more openness. Personality. This was a study done by Robin Carhart Harris, who's leading the field. We'll definitely be talking a lot more about him throughout this presentation. So psychedelics tend to improve openness and kind of liberalism, which makes sense because more of a conservative mindset is kind of more stuck on those rigid patterns. So the ability to kind of shut down and accept that other perspectives might be acceptable kind of opens up this liberalistic. And they did the study with cocaine and alcohol, and they found it did not cause those effects. There was also a study for nature relatedness, which is defined as I always think about how my actions affect my environment. They found that psychedelics increase nature relatedness. However, alcohol decreased nature relatedness. So when I think of environmental friendly, eco-friendly, openness, liberalism, kind of what comes to mind is Portland, which is maybe why we have such a strong psychedelic community here in Portland. So let's talk about high dose versus low dose. Microdosing is definitely a very interesting thing that is being kind of researched a lot more, and I want to cover that a little bit. So just a few definitions. So convergent thinking would be persistence over flexibility. So my idea is right, I need to persist to get this correct, versus divergent thinking is more flexibility over persistence. So in a high dose, you have an increase in divergent thinking, flexibility, a decrease in kind of this persistent thinking, and an increase in creativity. However, in a low dose, we see an increase in all three. So that might be kind of helpful to create this optimal balance. So again, microdosing. So the serotonin receptor, the 5-HT2A, might actually indirectly regulate dopamine. So this is kind of pushing dopamine towards the most efficient mind zone, kind of enhanced motivation, focus, reduces distractibility, procrastination, and rigid thought patterns and behavior, which might be kind of useful for maybe the treatment of ADHD, more light depression, and maybe that's why we see people in the Silicon Valley kind of using that. So I kind of made this graph right here. So we kind of have performance and function and dopamine levels. So you kind of see as the dopamine levels increase, you kind of get this arc. So this is kind of at the peak performance and function when you have kind of moderate levels of dopamine, which microdosing seems to do. However, at higher doses, higher levels kind of shut that down a little bit. So that might kind of benefit you depend on what you're trying to gain out of the substance, which might be a little bit more useful for kind of addiction treatment, where you really need to kind of shut down rigid thought patterns and stuff like that. But we will go into that quite a bit more. This was a very interesting research study that was done by Jeff Tarrant. He's a licensed psychologist, and he took 3 subjects and he hooked them up to EEG, which is basically measuring different brain waves that are produced in your mind. So what he did, again, he kind of looked at everybody. So this is subject 1, subject 2, and subject 3. And I won't get into the details of all the different brain waves, but as you can see, everybody's different. Everybody had a different kind of effect based on it. So the bluer means that there's kind of cooler colors are decreased activity versus warmer colors are increased activity. So this person had a little decreased while this person was firing. So very, very interesting that you would see this kind of different per person. But what he did was there's a national database with thousands of people's EEG data. And what they did was they averaged all of that data to create an average brain. So he compared these 3 people to an average brain. So then you see the blue is before they took the, this was done on, I forgot to mention, this was done on psilocybin. So this was on mushrooms on kind of a lower dose. So these people were microdosing on psilocybin. And we can see they kind of have higher levels, and what this is measuring is change in outlier z-score. And what a z-score is it's how different are you from the norm. So basically what happened in all of these is everybody's brain started to look more like this averaged brain. Even though everybody had a different effects on their brain waves, looking at it together, you can see everybody had a decrease in outliers. And he kind of described this as a balanced form of mental operation. that is more efficient. Because if you're getting a writer's block, you weren't having a writer's block before, you were writing fine, and then something kind of went in there to block it, so this kind of reverts back to your more original state where you can kind of get over that writer's block. So I want to talk a little bit about Robin Carhart Harris and the research he's done. He's the head of the Center for Psychedelic Research, Division of Brain Science, Faculty of Medicine at the Imperial College in London. And he's definitely leading the field with 97 publications currently on psychedelics. So he focuses a lot on serotonin because the 5-HT receptor, 5-HT2A, is very much a serotonin receptor, so psychedelics seem to have a major effect on the serotonin system. So serotonin is involved in neuronal plasticity. And what that is, it's the adaptability, the ability to change. So our brains naturally change with time and with experience. And that's called neuronal plasticity. So serotonin is very important to your context, which is environmental factors. So research in humans and animals has found that manipulating serotonin changes an organism's sensitivity to the environment. And they did this with things like SSRIs, which can increase the amount of serotonin that's in the synaptic cleft to kind of see how that affects an organism's sensitivity to the environment. And just to really understand plasticity, so if we did not have plasticity, we'd kind of all just have these traits, but plasticity allows us to adapt to our environment. So you can see all these are kind of converging on one thing that might be necessary for that environment. So there's a difference. Serotonin it's a complex relationship. So they found more enriched environments, if you increase serotonin levels, like SSRIs do, the positive effects of the enriched environment increase. It's a little hard to quantify in humans, but here's a kind of a good example from mice. I would describe this as a more enriched environment. They got little playthings, a wheel, stuff to run around, little ladders. However, a more adverse environment, serotonin increase is found to actually improve a negative response, which maybe is why SSRIs don't work well for everybody. And we got to think about who's really can afford to get the treatment, who can go into these kind of centers and obtain SSRIs. Maybe it's the people who are more in the enriched environments and they're receiving the more positive effects. But this is kind of a saying that it's a complex relationship that SSRIs kind of work like this machine gun and just sprays and hits all the receptors, versus psychedelics can seem to target one. So it's a little bit more precise and accurate. This was an interesting experiment called Rat Park. I'm not sure if anybody's familiar with it, but basically they had mice in cages and they gave them morphine and water, and the mice in the cages would drink the morphine until they died. However, what they did was they built a rat utopia. So there was a bunch of other rats to play with, cheese, wheels, all kinds of stuff, and they found that they actually barely drank the morphine. They kind of converted more to water. So it does kind of have a lot to do with what's in your environment and around that. And again, serotonin is involved in mood on the context of your environment. So increasing serotonin does not necessarily increase this positive mood. And so this really just hits at the importance of the context. So stimulation under psychedelic use. So psychologically, low-level associative learning increases. Extinction learning, otherwise known as unlearning, can also be increased. Flexibility in behavior and thought patterns is found to increase as well. And then physiologically, we kind of see neuroplasticity, which I described before, the adaptability, the ability to change increases. And then something called synaptogenesis increases as well, which is not the growth of which is not exactly it's kind of not the growth of new neurons, but it's more so the functional components of the neurons. So we can see here that this would kind of be neuroplasticity, a new neuron forming, versus here we kind of would see more synaptogenesis. So we can see a dendrite and an axon terminal forming. So it's actually able to build more of these functional components of the neuron when we have activation of these receptors. So again, this receptor is not just there for psychedelic use, it's very part of human development, and its highest expression is during key developmental periods. So there's less expression with older age. And this allows for more plasticity early on when learning is so important. And the brain kind of acts like a sponge, just absorbing these new experiences. Just some neuroscience 101, we're actually born with approximately 200 billion neurons. We love to talk about this at outreaches, but by age 24 to 26, we only have 86 billion neurons left. So we lose over half the brain cells in our brain. We love to go into the classroom and ask the K-12 students, who has more neurons? Who has more brain cells? You guys or the neuroscientists? And they'll be like, oh, the neuroscientists. No, it's you guys. The kids have more brain cells than we do. And that's kind of interesting, I always thought, but who can do more? A child or an adult? An adult can do more. So kind of a general way to quantify that would be, I like to tell kids about, think of the electricity as brain messages sending to different regions of your brain. So let's pretend we live in this house. We want to turn on this light bulb, and this is the power station. We try and flick on the light bulb, but it's not turning on for some reason. It's because we're not getting enough power to our house. And that's because, for some reason, the power company has all these power lines going nowhere that aren't really helpful for us turning that on. So we go ahead and we call the power company, and we're like, hey, we want to turn on this light bulb. Let's cut down on all these unnecessary wires. So the power company does that. By having only one wire going directly to where you need it, you're able to turn on that light bulb. And to kind of bring that out of the analogy, it kind of really is important on whatever you develop, whatever you're practicing, especially at a younger age, your brain will be like, oh, this is important. Let's send more connections to this area. Or if you don't practice something, oh, this might not be as important. We don't really need connection. So as we have that, we kind of see changes in how the brain is organized functionally. We see systems for specific functions increase with functionality and age, which allows us to kind of start doing, whether we practice musical instruments, reading, sports, and systems starting to communicate more coherently. And systems with different functions become more separate from one another. We kind of see a functional segregation, which is performance of functions with a greater degree of specialization. So you're able to kind of do those tasks a little bit better the more you practice at it and as you develop and get a little older. Especially if you're practicing playing a musical instrument through the developmental period, your brain will be like, this is really important. Actually, if you have a very young child and you kept them in a completely dark room until age 5, the brain would actually clip all the neurons to their visual system and they would be blind for the rest of their life because the brain viewed that as not being important. So it's really based on what you are doing. So again, back to under psychedelic use, you kind of see a breakdown of specialization, a system regression. So developmentally older brains start to revert to more looking like a developmentally younger brain. So you see a global integration increases and a local disintegration increases. And we kind of see a young versus old up here. And then this would be kind of all brain connections that occur. So kind of talking about how the brain is connected. There were two theories originally, modularity versus holism. In its raw sense, modularity means that each part of your brain does a specific thing and they don't talk to one another. Versus holism is any part of your brain is as good as another part. If you lose a part, another part will take over its job. Neither are totally accurate, although both, if you consider them both together, elements from both of these are rational, and both theories play a role in cognitive functions. And this is some pretty intense neuroscience where we're going to get into kind of mini columns and kind of macro columns. So a mini column is literally, if you were to go vertically down, there's neurons that are working vertically down. They all bunch together, and a few of them together create this column, a mini column. And then they kind of communicate through each other because all the neurons in that column are all doing the same thing, responsible for the same task. And then we have what's called a 1% neuron, which is connected to up to 100,000 different neurons. So these are kind of like your major connection nodes inside of your brain. So if we were to shrink this mini column down, we can kind of see these macro columns here. So it's a little hard to see, but this whole structure would be each one of these numbers. and that's how they kind of communicate to one another. So it might take a long time for go to 0 to 1 to 2 to 3, although you have regions of your brain that are directly connected, which is high closeness centrality, which they kind of jump over like that, which allows for instant connections on areas that are very important. And this is a side view of a cortical column, kind of what I was talking about. If we were to slice the brain and look at it kind of from this perspective, so these cortical columns kind of go down like that. And they're in different levels. Level 1, 2, 3 tends to send information to the upper wrinkly part of the brain, the cortex. Level 4 is very interesting. Level 4 is basically it's connected to the thalamus, so it's what sends those messages either up or down to different regions of the brain. So this is a very important part of the brain, these cortical columns, where psychedelics tend to take a big effect. And you can kind of see that there's like these little triangle looking ones, which is actually what I have here, which is a pyramidal neuron. So you can kind of see that they're very long, and they kind of start all the way at the bottom, and they spread really high up into the cortex. So these are kind of the ones that are connecting lower regions of the brain to upper regions, and kind of lower regions of the brain are responsible for your basic functions. So it's kind of your drives, your feeling, your perception, versus higher regions are more cognition and other things like that. So that might have something to do with why if somebody were to step on your toe, you were to feel something, and then you kind of get an emotion from that. So this is kind of a very important part of the brain, which we're also going to go into a little bit more further. And then these lower parts kind of go to the lower part of your brain, which I was talking about a few minutes ago. And we kind of look and when we're doing an MRI, we kind of look at these different regions and how they're connecting to one another. And that's kind of what allows us to build these pictures. And these kind of regions are called voxels, and they can either be big or small, depending on how we want to look at it. So somebody could look at just a few major networks, you could look at more networks, or you could look at all these different networks on a lower level. So back to the 5-HT2R receptor. Again, it's not just there for psychedelic use. It's really responsible for adaptivity, or the ability to kind of adapt to situations. And it seems to naturally turn on during periods of stress. And during times of stress, when we need to be thinking on our toes, especially when we need to be adaptable to survive, that is when the system engages, and it's useful and improves our chances of survival. So now we're going to talk a little bit about psychopathologies, which are like different mental disorders, and psychedelics and how they affect it. We're going to focus on current research that's happening, so MDMA to treat PTSD, Psilocybin to treat depression, and Ayahuasca to treat addiction. So both of these are actual research studies. The Ayahuasca to treat addiction proposal, this one is a proposal right now, so it's not based on an actual research that's been done on human subjects, but rather theory on how we know different regions of the brain communicate and affect each other. So let's talk about MDMA to treat PTSD. So this is kind of, a lot is brought by Mark Hayden, who's the director of MAPS Canada. He's an adjunct professor at the University of British Columbia, social work, nursing, medicine, public health, 28 years in the addictions field. And he has many publications on the issue of drug policy. So this is going to be kind of a lot of slides based on his presentation that I went to a while ago. So spirituality. Classical psychedelics offer a sense of generic spirituality. And what I mean by generic spirituality is Christians find the Christ, Buddhists find Buddha, and atheists find the entire universe. Whatever your belief system is, that is what is manifested. If you believe in the vine, you will find the snake in the vine. So this is incredibly helpful in the treatment of many issues. The disorientation of ego, when fundamentally one's self of sense changes. Very useful in the treatment of alcoholism. If it's one ego that is persuading one that it's acceptable to drink 26 ounces of vodka every night, that belief system needs to be disoriented. And classical psychedelics, they're useful for this. They're unconscious amplifiers. They help us to access material in our unconscious mind. the permeability between the conscious and the unconscious mind becomes less. We can dig deeply into ourselves and find stuff. And if issues are in our unconscious mind, we can find them and we can rework them. And it's also the portal effect, the wow, that was incredible effect. Like climbing Mount Everest or graduating from a university. It's a major sense of accomplishment, achievement, and transition. And that's useful in the treatment of substance use disorders, withdrawal and detox, depression and anxiety disorders, and possibly many more. So there's the empathogens, the connection with others. You have MDA, MDMA, 3-MMC, and these kind of bring the intense bonding, which is kind of what I was talking about earlier with the oxytocin release, versus the entactogens are more the connection with oneself. And that really helps on the therapeutic alliance. And this is the greatest predictor in success, no matter what orientation of psychotherapy you're doing. The alliance you have with your therapist is the greatest predictor of success. And these are alliance-building medications. They help one to reflect on oneself, reduce the fear of PTSD treatments. PTSD is like a tape loop that is buried in our unconscious minds. And as traditional therapists get close to this tape loop, there's a huge fear response. MDMA reduces that fear and allows them to connect and directly confront this tape loop. MDMA is a state-of-the-art treatment for PTSD. So I want to talk a little bit about Michael Mithofer's work. He did MDMA-assisted psychotherapy with MAPS, and he was discussing how it is different than other orientations of psychotherapy. And MAPS is the Multidisciplinary Association for Psychedelic Studies, who is currently pushing the field right now specifically for MDMA treatment of PTSD. They have several clinics all around the United States, and there's even one trying to be started here in Portland. So what Mark Hayden did was he went to the Canadian Military Health Conference in Vancouver, British Columbia in 2018. And what this was, where there was scientists pitching PTSD treatments to the military. And they claimed at best a 25% level of effectiveness. Literature says a little closer to about 10%. It takes years and the dropout rate is high because people don't want to re-experience their trauma, which is known as flooding or prolonged exposure. Sending a war vet to re-experience their trauma, to desensitize, is a very difficult experience. It's challenging. So Michael Mithofer demonstrated an 83% level of effectiveness, from about 10 to 25% to 83%. And if you're a researcher, you're going to claim they're lying. They're falsifying their data. At best, we see these increments increase by 5%. So a jump to 83 is completely off the charts. So MAPS took this information and they demonstrated in stage 2 clinical trials on a larger group of people with a more diverse population and many different therapists under the supervision of the FDA, a 67% level of effectiveness, which is absolutely astounding. Here's the paper. I'd be happy to send it to you if you shoot me an email, and you can kind of read a little bit more yourself about it. So now we're going to talk a little bit about psilocybin to treat depression. So depressive disorders, they're the leading cause of disability among American adults. So 16 million Americans had a depressive episode in 2015, but I would imagine that number is a little higher. It's kind of hard to gauge those things. So this was a research study done by Robin Carhart-Harris for severe depression. So he took 20 subjects, and they all had treatment-resistant depression. I hate that terminology. It sounds like they're resisting the treatment. No, the treatment's not working for them. And that is defined by the failure of 2 classes of medication. On average, these people experience their depression for 20 years. So I'm sure they've tried much more than 2 types of medication in those 20 years. I'm sure they've tried everything there was to be offered. So they used the QIDS, the Quick Inventory of Depression Symptomology, to kind of assess depression. It was just a 1-week experiment, so 7 days. And they had 2 psilocybin sessions, 10 milligrams, which is a light dose, and 25 milligrams, which is kind of a stronger dose. And in case you don't know your pure psilocybin off the top of your head, here's kind of a little graph to kind of better describe that from Arrowhead. So what they did was on day 1 of the treatment, they gave them 10 milligrams of pure psilocybin. Then they waited 1 week, so no doses were given. And then they gave them a larger dose, 25 milligrams, 1 week later. And then they did a brain scan the following day, an MRI scan, to see what was going on in their brains. 1 week later, every participant showed a reduction in their depression. 3 weeks later, 9 of the 20 participants were in full remission, which is absolutely astounding to consider these people have been depressed for 20 years, have all current treatments, all medicine has failed them. And in 1 week, 2 doses, half are in full remission, which is defined as very low levels of symptoms, if any. So this is the brain scan that they did 1 day post, the 25 milligram session. And kind of a major area that we like to look at with depression is the amygdala, which is loosely quantified as kind of your emotion center of the brain. It's very much responsible in the fear response, the flight or flight response. And they found that the blood flow decreased 1 day later, which is associated with a decrease in depression. But interestingly enough, they found the reactivity of the amygdala increased. And what they do to kind of assess that is they show people in a very common experiment happy and sad faces, and they see how the amygdala responds to both the happy and the sad faces. And what they found was it increased responses to both. So it increased the response to the fearful face, but it also increased the response to the happy face. Now we got to remember, these people have been living with their depression for 20 years. So they were very used to kind of feeling these negative emotions, but possibly the happy feeling, finally the ability to feel that happiness might have made all the difference for them. That is something they might not have experienced in those 20 years, which can be kind of quantified as the acceptance of all emotions, both good and bad. Now, SSRIs, their goal is to reduce the reactivity of the amygdala, to make you less reactive, and the idea behind that is you'll be less reactive to the negative fear faces, but it also makes you less reactive to the positive faces. And this showed that psilocybin is the polar opposite effect of SSRIs, which I find absolutely interesting. And then we also talk about the default mode network, the good old default mode network. And then they found the functional connectivity of the default mode network, so how it connects to different regions of the brain, increased. So they measured 5 weeks later, and they found that 9 of the participants, the 9 who were in full remission, had spatial increases in the default mode network. And that's a similar outcome that we see to electroconvulsion therapy. But if you ask me personally, I'd much rather eat some mushrooms than get shocked. And that could be associated with the clinical outcome. Because, I mean, the 9 people who went into remission were the people who had spatial increases in the default mode network. So that's a very good indication that this has something to do with the clinical outcome. So the subjective effects, so cognitive flexibility increased, well-being and optimism increased. But it was all related to this mystical experience. So they found that when you have more of a mystical experience, psychological well-being increased and depressive symptoms decreased. And this was also more research done by Mark Hayden as well. So what a mystical experience, it's one of the greatest predictors of success in the treatment of psilocybin. And it is the amount that people have in a mystical experience that seems to be connected with these positive treatment outcomes. So it's often highly correlated with the success of the outcome. And people with the greatest mystical experiences find the greatest effects from these treatments. But how do you measure mysticism? So the most recent one is the 30-item revised mystical experience questionnaire, MEQ30, developed by Griffin et al. In 2000, or the validity was tested in Griffin et al. By 2015. However, these questionnaires have been around for a while. We've been using them for about 50 years. These are all people who have used them in their research papers. The most frequently used is the MEQ 43, which has 43 items, although the newest one is the MEQ 30, 30 items, a little less items to test on the participants. This is a validity paper to test how well it's able to measure that and internal consistency. This is a slide from hell, but this is a confirmatory factor analysis. It's a statistical measure to build what's called a latent variable. And what a latent variable is you can't measure mystical or mysticism, but they found all these questions that statistically load onto this one variable. So you can measure things like mysticism, positive mood, acceptance, inevitability, based on these questions given the statistical formula. And Cronbach's alpha is a statistical measure of internal consistency. And this is kind of a little table up here. So if it's 0.9 or equal to 0.9 or above, it has an excellent level of internal consistency. 0.8 and above is a good level of internal consistency. Normally you see papers kind of in this range. It's pretty rare to see stuff up there in the excellent. So they measured each of these latent variables. They found mysticism, 0.97. So that's excellent. Positive mood, 0.92, excellent. Inevitability, 0.9, excellent. Transdimensional space and time, pretty good, pretty good. So this is really saying that these questions are measuring what we think they are, and we're actually able to gauge what is responding from this. Although I find these questions to be very interesting. Oceanic boundlessness on a scale between 1 to 10. Feelings of peace and tranquility on a scale between 1 to 10. The belief that you've contacted truth on a scale between 1 to 10. The inevitability of it. Can you find words? No! On a scale between 1 to 10. So this is a very interesting study that was done. They took subjects and they gave them the MEQ, and they measured basically mysticism. There was it was a pre-post-test blind test design study. So they measured them before they took the substance, after they took the substance, and they did not know what they were taking. So there were two groups. One took psilocybin, the other group took Ritalin. And so they did to measure that as kind of the placebo or the control group. And they measured 2 and 14 months later, they re-administered the mysticism. Not the drugs, they only gave the drugs one time, measured before, and then they measured later. And they asked the participants about positive mood changes, an increased sense of spirituality, how personally meaningful and spiritual this experience was for you. And what they found 14 months later was the numbers went up. So here we see at screening, before they took the drug, it was kind of low. 2 months, major increase, but 14 months later, even a slight, even greater increase. And here's another slide from hell. I won't go over that too much, but so this is a groundbreaking study. Can you think of any intervention a psychologist, psychiatrist, or a doctor does to anyone that you wait 14 months and remeasure, and the number goes up from a single treatment? And when administered under supportive conditions, psilocybin occasion experiences, like spontaneously occurring mystical experiences that at 14 months follow-up were considered by volunteers to be among the most personally meaningful and spiritually significant experiences of their lives. So when they asked, why did this treatment work for you? People said they went from disconnect to connect. The avoidance of difficult emotional experiences to the acceptance. And they found that CBT and other antidepressant treatments kind of reinforced this disconnect because it didn't work for them, or maybe they were handed from one therapist to another. Maybe it had something to do with a therapeutic alliance. They didn't feel like they were really bonding with their therapist to a great degree. So some kind of metaphors for that would be a calm after a storm, a broader perspective, kind of a reset. Flexibility, kind of like a mind lubricant, and recognition. And I like to kind of describe this as the acute stage. So when you're under the influence of psychedelics, it's like kind of molten metal, might not be the best time, or like hot metal. That's not a good time to kind of apply treatments to it. But the afterglow, kind of this after period that we see, is more moldable. So this would be kind of where we want to apply these treatments. So ayahuasca to treat addiction, and this is a proposal. So this is not actual research that has been done, but it's based on different brain structures and how basically the mind works psychologically and physiologically. So substance use disorders, they're bad. They present a global, enormous global epidemic exceeding $500 billion every year. 22.6 million individuals suffer from them. 2.5 million from alcohol alone in the United States, and over 200,000 fatalities from illicit substances. So they're associated with kind of a downregulation of dopamine receptors, and that's associated with kind of addictive behavior. The efficacy of dopamine is reduced when you have less receptors. So that means less pleasure from daily functions, from kind of like waking up in the morning, from eating food, from sex, from just like anything that, like hearing the birds in the morning kind of reduces that pleasure from those experiences. So here we have a beautifully drawn synapse. So here would be the axon terminal up here, and the neurotransmitters are stored inside of there, and then these would all be receptors. And they're G protein receptors. So basically what happens is a little protein vesicle comes down carrying the neurotransmitters. They're released into the synaptic cleft, and they bind to different receptors. So this might be a high dose of a stimulant, possibly, that could release a lot of dopamine, and that causes G proteins to be released inside of the cell. And the cells don't like that. Too much activity, they want to protect themselves. So what do? They cut out a bunch of receptors to help save themselves. So that means if you're not using the substance, you're just trying to do a normal activity that might give you pleasure, dopamine will try to be released, but there's less receptors for it to bind to. So you kind of see this reduction in pleasure from that. So higher levels of dopamine is associated with more addictive behavior, versus lower levels of dopamine are associated with kind of withdrawal and dysphoria. So this is kind of the hypothesis of the treatment. Broke it down into 4 different kind of sections: the biochemical, physiological, which are more basically in the brain and related to structures, versus the psychological ones, which would be, again, just psychological, and the transcendent. That's just kind of a graph that I built to kind of describe that a little bit better. And I specifically thought it would be good for methamphetamine because that's very much associated with dopamine, even more so than cocaine is. So this is the biochemical theory. Basically, DMT, which is a major component of ayahuasca, is, as we discussed earlier, a serotonin 2A receptor agonist. So it binds to it and it turns it on. And that tends to release glutamate. So you've heard of many neurotransmitters that I've described, such as dopamine, serotonin, acetylcholine. But the most common neurotransmitters in your body are glutamate and GABA. And glutamate turns things on, and GABA turns things off. So they're the main workhorses of the brain. And all these other neurotransmitters tend to affect the way that either glutamate or GABA is released. So GABA is found to inhibit dopamine, so it prevents it, it turns things off, so it turns off the flow of dopamine. And you have a MAOI that preserves the remaining dopamine, and that stabilizes levels of dopamine in this motivation or reward center of the brain called the nucleus accumbens. Very important structure in the limbic system. So again, this is the mesolimbic pathway that dopamine uses to basically send chemical messages out to different areas. The VTA is in your brainstem, which tends to house a lot of neurotransmitters, and that kind of sends messages out to the nucleus accumbens and to the entire limbic system. And the prefrontal cortex, which is very responsible in kind of cognition, and the hypothalamus, which is very responsible for basic drives, like food, sex, water, just basic things that our biology believes necessary for either survival or continuation of the human race. So here we have a brain. And again, we have these kind of neurotransmitter sections down here. The RAF-IA is very important for serotonin. And then here is kind of where the limbic system would roughly be. We have the striatum, and the nucleus accumbens is kind of right down in that section. And what happens is we have a very long pyramidal, which is the ones that I have here. So these really long ones that kind of stretch all the way up there. And we also have a glutamate neuron, a glutamatergic neuron, which turns things on, and that kind of comes back. So it kind of creates these functions, or this kind of circular network. So DMT tends to activate in higher levels of the cortex, where I kind of described earlier. There's a high level of the 5-HT2A receptors up here. So here we have my beautifully drawn synapse again. And let's just say this is this could be, you have 15 different ones, so let's just say these two are the 5-HT2A ones. So DMT will actually rush in and bind to those receptors. And you might ask, and then cause intercellular changes. So you might ask, why does DMT bind to it? Well, those receptors are meant for serotonin. So this is serotonin on the screen. And let's bring in DMT. Well, that's a pretty close fit. So it actually is essentially a very similar structural chemical to that, so it can bind to it very well. So then you would have an electrical message go down, kind of travel to this neurotransmitter section down there. And here's a kind of a close-up of that. So you'd have the glutamate neuron, an electrical signal will kind of come down, release neurotransmitters into the cleft, and then you have this GABAergic inner neuron, which again turns things off. So the message will continue going, and then the GABA is sending a canceling message to this dopamine neuron, and that kind of goes up, and as we can see, this would be the dopamine neuron. It'll send that inhibitory message, the turning off message, to the nucleus accumbens, or the reward center of the brain, reducing the levels of dopamine that are in that region. Although, as we kind of mentioned earlier, just reducing the levels would kind of promote withdrawal and other kind of these dysphoria, not a good feeling, which is why it's pretty hard to kind of revert from substances. But ayahuasca, the DMT helps to reduce the levels of dopamine that are specifically in that region. However, the MAOI kind of preserves that remaining dopamine. So again, you have the synaptic cleft here, axon terminal, dendrite. So let's say dopamine comes down, it's released, it binds the receptors. Now, as I mentioned earlier, they don't enter the neuron. The neurotransmitters don't. The G proteins are what breaks off and goes inside. So they actually get sucked back up by this reuptake transporter. And inside there you have monoamine oxidizers, and they basically break down the neurotransmitters. They help to reduce them. So if you have an MAOI, they inhibit that and allow for the dopamine to be repackaged. So you're reducing the amount of dopamine that's here in the synaptic cleft that can bind to other receptors, but you're increasing it in this region of the neuron. So that could be kind of quantified as the ability to kind of, when you need the dopamine, like, or when you naturally would release it, like when you're eating food, and waking up in the morning and hearing the birds, you'll actually have this pleasurable experience. So let's get back to, let's say, this damaged neuron that had a lot of the receptors reduced. So your brain over time will realize, hey, I'm not getting enough activity here. So it'll actually send G proteins down and it'll create more receptors there. And so your brain has a very unique way of repairing itself. So even if you remove these receptors, it's not the end of the world. You can come back from this. And that allows more neurotransmitters to bind and kind of revert more to a quote-unquote healthy brain. And we have the psychological. So it enables the processing of significant conscious and unconscious experiences, helps to resolve trauma. And this really has a lot to do with kind of this lack of social integration and stress that is experienced. Not to go too Freudian, but we have the ID, ego, and the superego. And there's something called the three-horn dilemma, where you're trying to satisfy you're trying to satisfy them all, but it's very difficult to do so. So you're probably satisfying two at a time. So ego dissolution, the death of the ego, allows you to satisfy your ID and your superego. And I like to describe these structures. The ID, if you think of two kids playing with a truck, the ID would be both the children want to play with the truck. It's your want, it's your desire. The ego is a little different than a literary definition. A psychological definition of ego is how do you feel connected with your environment? So a child might be thinking, I want to play with this truck by myself. Am I going to get in trouble if I steal it? Is my mom going to yell at me? Versus the superego is more the morality of it. Do I feel okay with stealing this truck from my friend? So this kind of allows for the shutting down of the ego to allow these connections, and the three-horn dilemma is solved. And then there's the transcendent idea. The idea that can change a person's beliefs, values, and worldview, promoting a connection to their environment. And in our society, we have this very strong preoccupation with individualism. I moved 3,000 miles away from my home and my family to go to school. And in a collectivistic culture, you probably would never do that. It's about a family first. But here, in our individualistic culture, it's totally okay to take ourselves first, you know? And that kind of leads to this social disconnection, which can very much make you feel disconnected with your environment. And it's not necessarily just a disconnection with the outside world. You could feel disconnected with your internal world. You could feel like, wow, the world's falling apart and there's nothing I can do about it. You might have a sexual orientation that's not appreciated. There's many different things that could kind of lead to you feeling this disconnect with your environment. And again, the lack of social connection taps into the same neurological system, the alarm system that responds to critical threats. So if somebody came at you with a weapon, that same part of your brain is responding to a lack of connection because it's dangerous. If you think about kind of back in the day, maybe when there were saber-toothed tigers, if you were on your own, you were in a lot more danger than if you were in a big group of people. And your brain is kind of trying to say, hey, we need to connect and build a connection with other people. But similarly, social connection actually taps into that reward system. So I like to go to schools and tell kids, like, you don't need to use drugs, you can just make friends. Like, it's the same network. Okay, so that was a lot of information that I threw at you, but let's talk a little bit about the dangers of psychedelics. My personal favorite, girl gives birth to a frog, and doctors blame LSD. And then we might have seen that guy before, drugs are bad, okay? So I want to talk a little bit about psychedelic safety. This is a really interesting chart. So this is dependence level, so how addictive it is. And this is the active dose versus the lethal dose. So kind of how much do you take before to get high versus to where it can actually kill you. So we see down here, LSD and psilocybin have a very low dependency, and you have to take massive amounts of it before it would be fatal. Compared to how much produces a cognitive effect. And I always think it's interesting that all these ones up here, kind of in the more higher levels, so very high addiction potential, and the dose that gets you high is very close to the dose that can kill you. These tend to be the legal ones, which I'm kind of confused about, honestly. So I want to talk a little bit about methods to reduce the dangers of psychedelic exploration. So I heard I recently went to a MAPS Zendo project, which is a harm reduction training session. And this is kind of the main points that I got from them. They said, do your homework and clean your room. I really like that analogy, and what they mean by that is if you have a lot going on, if you have a lot of kind of responsibilities, like that specific day might not be the best day to go on a psychedelic exploration. It should be a day that you can just fully commit to that experience without outside distractions. And they described it as kind of being like a bridge, the psychedelic journey. Once you get on, you don't really get off, but you rather travel to the other side. And trying to hop off in the middle or trying to stop the trip could be kind of quantified as jumping off the bridge. And of course, be safe. Don't drive or operate a vehicle or heavy machinery. It's always good to have a trip center present who's preferably not intoxicated or not heavily intoxicated. Be in a familiar area and have a safe space that you can kind of go to if you need to be alone for a period of time. And always, 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 if at all possible, test your substance. There's some places like Dance Safe, which is a really cool website, and you can buy kits on there just to make sure you're consuming what you think it is. And we're going to go over the dangers of what happens when you consume something you don't think it is in a minute. And it's always good to remember that difficult is not the same as bad. Sometimes these medicines will amplify our unconscious mind of something we need to work through. That can be an amazing experience. Like if you see somebody at a concert and they're running around screaming at the top of their lungs on a psychedelic experience, maybe it would be a good idea to ask them to calm down, but you don't really know their backstory. Maybe they've been repressed their whole life. Maybe they've been told you're not supposed to talk loud. You're not supposed to say who you are. And that ability to kind of bring that out was such a meaningful experience to them that really helped them to kind of feel like rejuvenated almost. And if you're a trip sitter, practice soft gazing rather than directly staring them down in their eyes. It might feel a little confrontational if that happens. And kindly remind the person of their surroundings. Hey, this is great, but let's get off this public road. Or I love your energy right now, but maybe let's tone it down a few levels so the neighbors don't get bothered. And only offer what you can provide or you have around, like a blanket, food, water, a hug. It's not good to make the mistake of being like, I'm here, whatever you need, just let me know. Well, I need a rainbow unicorn that has magical wings. And I'm like, oh, I don't think I have that on me. And then help to prevent situations that may lead to future regret for the person. I want to call my boss and quit. I want to call my mom right now. Hey, that's great. What if you write down how you're feeling right now, and if you still feel the same way tomorrow, give them a call. And if the person is having trouble on the come down, like they can't sleep or it's just going for too long, art's a really useful tool. Like maybe give them a few coloring pencils and a notebook, just kind of a way to channel that energy. I've been told that people have been like, oh, I can't sleep, and then they just do a little bit of coloring and then 20 minutes later, they're knocked out like dead asleep. So that can be a really useful tool. And then always remember, if the situation is out of your control, obtain help and be the spokesperson for that person between the help services and that person. Make sure that they're safe. There's a lot of evidence that shows restraining people can be very negative under the influence of a psychedelic, especially kind of what we were talking about earlier. Just being able to let that out might be a good experience. But if they're putting themselves in danger or other people in danger, there's a place where to draw the line, and that needs to be recognized. So again, kind of just a recap, safe space, you're sitting a sitter for them, not guiding them. Through, not down, and difficult is not the same as bad. And through, not down is like, we don't put people down for kind of exploring or kind of going through, like, you know, people sometimes get naked or all kinds of different things. And it's not a good idea to kind of scold them for that, but to kind of go through with the experience and kind of just be their barrier between the outside world and what they're experiencing in their head. And this was all from the Zendo project, which is, again, MAPS harm reduction training program. Okay, so this is a very interesting case study. It's about a 5-minute audio clip. It was a male a 17-year-old male who took a research chemical, 2,5-I, with a group of people, and in the group of people, he was the only person to have a very, very bad experience. And again, with research chemicals, we really don't know the long-term effects. We don't know what interactions they will have. So he took an acne medicine called Accutane, which is actually a black box medication. It's very dangerous. It has like a suicidal risk to it. He took it in the morning and then proceeded with this psychedelic trip at night with his friends. And he was under no impression that there would be an interaction. That's kind of the dangers that you have with that experience. So this is a 5-minute clip from him.
Yeah, so I woke up,
Is the volume okay for everybody?
And then about 12 hours later, I decided to try this new drug with my friends called 2,5-I. And I didn't know much about it. I just heard it was like stronger than acid, and I've taken like, you know, psychedelics regularly, so I thought I could handle it and stuff. I remember when I took it, it made my whole face completely numb, and it just had this really gross metallic taste, really like synthetic tasting. But yeah, I didn't think much of it. Moments later, I immediately started feeling effects like vibrant colors, jitteriness, like just anxious. And my friend was asking me, "Oh, are you good?" I'm like, "Yeah, I'm all good. I'm just really high, like really fast." And like, this was weird because normally psychedelics, in my experience, they've taken like 45 to almost like 90 minutes to kick in, and this was almost immediately. So the visuals start getting more and more intense, and I begin to panic. I then start to see like cop lights in my house, and then I get paranoid, like, oh, am I being too loud? Like, they must know. So I just tell my friends, like, I'm just gonna crash, I'm just gonna go to bed. You know, I thought sleeping would make me feel better. So I got under the covers, and that's when everything got really crazy. I just kept waking up confused, like each scenario more bizarre than the last. And I remember just not being able to come up with a coherent sentence or thought, and everything was all jumbled up, like a word salad of real and not real words that had no relation towards each other were just forming in my head, and I couldn't communicate to my friends that I was messed up, and I thought I gave myself like permanent brain damage, and that's when I started getting really confused. And yeah, that went on for a long time, and then I heard this really loud static noise that just kept building gradually. It just kept getting louder until it filled the room, and I just felt so much pain and just scared and just terror, and just I thought, this is the end. This is what dying feels like. I'm dying right now. And I felt really guilty and really sad because at the time I was like 17, and I was like a month away from graduating, and I was thinking, oh, how could I go out like this, and you know, how could I let my dad down? And and I think somewhere after that, that's when my friend tried to calm me down, and he tried to offer me a banana, and that's when I got really religious at that point, because I thought I was dying, and at that point I went back to thinking of Adam and Eve, and it's really strange, but you know, when Satan was, you know, trying to get Eve to eat the apple, I kind of thought in the same sense. So I grabbed the banana and I attacked him with it, and it took about like 3 of my friends to take me down, and that's kind of when I lost myself and went to another void, and then when I came to, I saw my dad, who now I know that wasn't my dad at the time, but yeah, he was there, and he was crying, and he was like, my boy, what happened to my boy? And I felt so guilty. That's when I felt just like complete shit. I just like, yeah, I just you know, it's really hard to see your parent cry, especially over you and your dead, and yeah, I saw the whole, I saw the paramedics come in. I got zipped up in a bag, and I saw myself from like a third bird eye view, and it was really real. And I truly thought that I had died, and I just messed everything up, and that was it. And then, that I'd just be known as the kid who died at 17 years old. And that went on for just like, it felt like eternity, and it just, yeah. Sorry, I lost my train of thought. Oh yeah, and then I got naked because I thought that's the only way that I could get raptured. That at the time it made sense, but yeah, I got naked and I jumped out of the window, or at least I tried to. I got halfway out, and the other half of me naked just kind of dangled in the living room. And eventually I just came to. What helped was towards the end, I think my sister was playing the guitar, and it was so funny. I was so high and so gone, and no one could get through to me. But my sister playing guitar, singing like a Bob Marley cover, but like she put my name in it. Like it was like "Nico, Don't Cry" or something like that in the Bob Marley type deal. But that was the only thing that broke through and made me feel comfortable, which was so bizarre and strange. And I thought I was at my own funeral, my own wake. Yeah, it lasted for at least, they said I was like that for 5-6 hours, screaming at the top of my lungs, and my neighbors, laughing, and my sister, defending me, and yeah, that's it. That's all I got. Thanks a lot.
So these are very powerful substances that we have to, very much be aware of, and if we're experimenting with chemicals that we don't know what kind of interactions will occur, risks like this could happen, and might have been a good time for them to basically call in, like, we need help for this kind of a bigger level. This might be out of our control, especially if somebody's doing dangerous things like trying to jump out of a window or swallow their own tongue or other things like that. Let's see. So 2,5-I is a psychedelic hallucinogen that was used to just research the 5-HT2A receptor initially, and then it got broken out of the lab. It was discovered in 2003, so it's really new, and it's the first full agonist, so it turns on of the 5-HT2A receptor, and it's 16 times as potent as 2CI and the 2C series, so it's an incredibly dangerous substance. Basically, there's a huge recreational risk that comes from pharmacological and both behavioral toxicity, as we heard from that subject. And it's a relatively new substance, so we barely know anything about it. It's not like LSD. It can be fatal in very low doses. It causes what's known as blood platelet aggregation. So the platelets will actually stick together, which can clog an artery and cause heart failure. So it very much can kill you in recreational doses. So it's kind of similarly sold as acid sometimes. It's available on things like the Silk Road, so people can buy it for pennies on the dollar, and some pretty heinous people sell it as LSD. So this is kind of really important why you should test your substance and know what you're consuming before you consume it, because you don't want to get stuff like this. And as I mentioned, Accutane, it's an acne medicine for severe acne, and there's a black box warning to it, so it has a suicidal risk, and it actually was interestingly found, I did a little research on it, Accutane and 2,5-I affect the same receptor. So there was a direct interaction that occurred, and that's kind of the danger of using these substances, is we don't know the effects that will kind of occur with them. So you want to be a psychedelic researcher or clinician, and this is the little last section of my presentation. So if you're interested at all in researching psychedelics, there's a few good things that you probably already know, but it'd be good to kind of be stated. They're heavily stigmatized. There's no infrastructure that you can channel your energy as of yet. No research graduate programs, student groups, scholarships, and few professors are willing to provide mentorship. There are a few colleges that actually do some research on it, but they don't not so much offer it to students. It's more the people who already have their PhDs and other things like that, although they might be willing to take on a few, but it's very, very rare. And at like 2 or 3, like Johns Hopkins, New York NYU, a few other places like that. So why would you be interested? Is it because psychedelics are novel and cool? Well, you're going to find it pretty boring because this research takes a very long time. Is it because you had a mystical, life-changing experience on a psychedelic? Well, you don't need to be a researcher to continue having those mystical experiences. Is it because you're frustrated living in a culture that tramples individual freedoms? Well, there's kind of other ways to be an advocate. Or is it because you're motivated by a genuine curiosity about these substances and a wish to apply the tools of modern inquiry towards understanding their properties? Perhaps you appreciate that scientists have found psychedelics valuable tools for formulating great discoveries and wonder how that can be so. Maybe you know the discovery of LSD was sparked interest in the entire serotonin system that prompted the entire explosive growth of modern psychopharmacology that continues today. Possibly you contemplate what other wonders may lie hidden in the closed box of psychedelics. So if this is your interest, it's probably right for you. But are you willing to accept that your unconventional interests may lead to professional isolation or even ostracism, and that the time-consuming navigation of these layers of red tape endemic of psychedelic research will inevitably slow your publication rate and your promotions compared to your peers? You will watch your peers accelerate much faster than you who are researching more conventional things. Are you aware the total lack of government and corporate support for such endeavors means that you will never be rich? You may in fact eventually land up in jail on charges of one sort or another. I mean, if you're willing to take these risks, as an undergraduate, get your degree, lie low, infiltrate the system, educate yourselves about psychedelics, start a student group, volunteer. Like, for example, I love to go to Northwest Noggin and talk with older kids, high school students, about psychedelics or specifically whatever substances they have. I don't go in there and tell them this is a psychedelic. I ask them, what do you want to know? And if a kid asks, I took this drug once, can you tell me about it? I'm more than happy to kind of share that with them. Oregon Psilocybin Society also has some pretty amazing volunteer positions as well. Write letters to your congressman. And as a graduate student, your first stop could be the Heffler Research Institute, which has a whole list of allies in the international academic world. And if you want to go the PhD route, neuroscience, neuropsychopharm is very useful. Experimental psychology is good as well. Clinical psychology is very good if you're planning to kind of apply these directly in kind of laboratories or research them. Also psychoanalytic, which definitely deals with the unconscious mind. So that's a very kind of useful tool to kind of looking at bringing out the unconscious mind. It's not just for MDs anymore. And cognitive science or just pure science is a really useful field. So any of these would kind of be a good way to launch yourself into that field if you're willing to take on those risks. But there are many paths to becoming a researcher, but the most important one, but what matters most in the end is that you obtain success and satisfaction on a personal, professional, and spiritual level, while at the same time remaining true to yourself and to your beliefs. If you want to be a psychedelic clinician, explore training programs and degrees in transpersonal-oriented psychotherapy. That seems to be a very key component of many of the applications that psychedelics are being used under. The California Institute of Integral Sciences, a certificate program in psychedelic-assisted therapies is offered over there. MAPS has an MDMA therapy training program. It's quite competitive though, but there are definitely ways to kind of go through there. Attend a Zendo project training session and join as a peer support volunteer at festivals such as Burning Man, or start your own practice as a holistic therapist. Above all, practice self-care and stay within the law to avoid unnecessary consequences. We hope to change the laws soon, but if people are breaking them, that's not going to help. Thank you. Nice some books. We have another 20 minutes or so for time for Q&A if anybody has any questions.
Yeah, you can take that off.
Okay.
So with MDMA, I've heard a lot of people talk about the come down not always being pleasant. So if you have access to something like Cymbalta, would that help?
There are definitely ways to kind of like help reduce the come down. Melatonin's a very kind of interesting way to kind of help like improve serotonin levels inside of there, because actually serotonin itself is converted into melatonin. They're very similar chemical. 5-HTP is also a really good way. I prefer the more natural things, because if you start getting into more of the pharmaceuticals, again, you can kind of hit interactions, but there's many kind of different ways. But the best way to avoid kind of long-term come downs on MDMA is to respect the drug, to use it in small doses over a period of time. A lot of times you hear kids consuming 8 or 9 ecstasy pills in a night at one of these parties, and that's just going to really kind of mess it up. In a psychedelic therapy session with MDMA, it tends to be quite lower doses compared to, off the top of my head, I want to say between 75 to 125 milligrams, but somewhere in that range. And then, so it's kind of lower levels to help kind of reduce that over effect. Yeah, we got Ben right after.
So I'm doing treatment-resistant depression with ketamine and LSD right now, and I found success with that. The LSD I've switched to microdosing, and I'm, since it's basically undetectable, I'm wondering if it's the right tool for the job or if psilocybin would be instead. And I'm wondering chemically how do these compare as far as addressing symptoms, how do they compare?
They're very, very similar. I believe that LSD, this is kind of with anecdotal knowledge, that would kind of work in a similar sense to serotonin because it both affects very similar receptors. It's just the research seems to be kind of pushed towards psilocybin, but that does not mean that LSD would not work in the same way. So, it's kind of whatever works for you kind of a thing. So if you're kind of interested with maybe experimenting psilocybin a little bit, see if one works better than the other, that might be a good way to go. I have a feeling we'd see somewhat similar results with LSD if it were to be tested, but the push seems to be towards MDMA and psilocybin currently.
As a parent with kids who experimented with X and stuff, I was always very fearful about that.
Absolutely.
And the way that I told it, and I thought that I had read that you can blow out your serotonin receptors and they don't regenerate. And I always told them it's like all that fun they were having is like taking a cheese slicer off your capacity for happiness the rest of your life.
So
but it doesn't appear, in this story. And you mentioned about the possibility of receptors regenerating.
Exactly. So we have upregulation and downregulation. If there's not enough activation at a neuron, it'll create more receptors. If there's too much, it'll remove those receptors. But as long as the neuron is still alive, it will come back. And kind of the dangers that we see with X use would be if there's methamphetamine directly inside of there. Because methamphetamine is so powerful on the dopamine receptor, it's one of the few drugs that actually kills the neuron. So if you get the neuron to be damaged or dead, then upregulation, downregulation won't affect it. And you see there's different ways that people consume that. Like sometimes people just take the pure powder, MDMA powder. Otherwise, people will have these pressed pills, which are incredibly dangerous, and they can have any kind of other drugs mixed in there, including methamphetamine. Sometimes there's no MDMA in there, and it's just methamphetamine pills that people are taking. And long-term exposure for methamphetamine is one of the few drugs that can actually just kill these neurons permanently. So everything is kind of within moderation and stuff like that. So lower levels will definitely.
Can they test for that neuron health then?
They can. So they can basically, they do PET, which is they inject a drug that binds to the neuron, and then they tell people to do some kind of dopamine releasing activity, and then they look to see how many of that drug is still bound to the receptor. So if they see like a lot of them are gone, they'll be able to tell there's some significant damage going on in there. So they can look inside of the brain and kind of see that as well. I think we had one question.
Just to sort of follow up on what he was saying about ecstasy. I've never tried it before. Had a couple chances, but it never happened. But I remember watching this thing on back when MTV was a little more informative. They had this thing about they had like these these sort of like these half-hour sort of specials, and they want about like drug usage. And there's one where this woman, she was like this young adult, and she ended up like having to move back home because she had done so much ecstasy. It like sort of like really messed with like her emotions. Like she was really having a hard time sort of like being able to function in your daily life. So maybe like the stuff she was taking was like a pill and had like a lot of methamphetamine in it or something like that.
Absolutely. And I mean, even everything is with moderation. So if you're taking massive quantities of MDMA, it can also cause damage. It's just much less common unless there's methamphetamine kind of mixed into there. But everything with moderation, for example, you can actually die from taking too much LSD. You need to take a lot, but it can be fatal. Anything can be fatal. If you can die from water. If you drink too much water, that can kill you too.
Yeah, that's happened. Yeah,
absolutely.
Yeah. So I actually had a question. One of those interested in coming to these things because learning about like how much like constitutes a microdose. Because I've definitely done like macrodosing, but I mean, and I might have done microdosing, but I'm not sure. Because I've heard some people say like, oh, it's not really perceptible.
Like 10 milligrams might even be kind of getting up there, so kind of like lower in that range, and everybody kind of perceives it differently. Like in that initial experiment that I showed you with the 3 different people who had the EEG caps all on, they all took the same dose of psilocybin, which was a microdose. Some people felt almost nothing, but one person actually started seeing colors and experienced a psychedelic trip. So everybody kind of has a different level of experimentation, and especially if you're consuming the raw mushroom, it's harder to gauge exactly how much is in that raw mushroom.
Yeah, how much is like I mean, I know it can differ from different types of mushrooms you're taking.
Absolutely, yeah.
But and I've had ones that I've like some I get like good visuals, and others are just like get more sort of a body high. So I mean, but on average, like how much like sort of active like psilocybin do you get per like say like a gram of material?
It's so variability. It would be kind of hard to say that exactly. I could give you kind of like maybe a ballpark of like 5 milligrams to 15, but again, that's super. That's
5 to 15 per 1 gram.
Ballpark. Again, yeah. I mean, if you have a really, really potent mushroom, then it could be kind of high. I've heard of people being able to have full trip experiences off of like 1.5 grams of mushrooms, but it's how they're cultivated. Some people just pick them in the wild. Some people cultivate them in fish tanks and are able to control the environment. And it also kind of depends on when you pick them. At a certain time, you're going to have higher levels of psilocybin. So if they're picked at different times, 10 to the highest one, you can kind of tell if the cap is open. Then there's probably going to be a little bit less than if it's kind of closed and more of a button shape.
So if it's more open, then it's a little higher.
Lower. So the button shape kind of retains a little bit more when it's in that state. But again, it's super hard to kind of dose. But I hear people kind of start, it might be safe to start off with like even as low as a half gram. See if you don't feel anything, maybe try a gram. Kind of continue to go to where it feels right. And other people kind of approach the microdose differently. Some people don't like or don't even want to feel any kind of an experience from it, while other people want to feel just a little something from it. So everybody kind of has a different variability for that.
Yeah, so the spirit they're trying to microdose, they're doing the 10 milligrams of active psilocybin.
Yeah, so that's how much they took in there.
So that'd be like ballpark of a gram, but like I said, it can differ because of different types. And
although, if we go all the way back,
I'd also just like to add on, there's so many different factors too. How much you ate that day, how much you've been drinking, sleeping, will affect your experience of it.
Also different pathologies. If you have like bipolar, borderline, it's not usually the smartest idea to take lots of psychedelics.
Also, Arrowhead is a really great resource. This is, I lost it, but they have this was one of the slides that I had up earlier, and they have a whole, like a threshold dose they say is 3 to 4 milligrams. Light is 4 to 8, so common is 16 to 20. So actually the 10 milligram range is kind of in that common one, so probably like they're saying a light dose is between 4 to 8. But remember, that's like so much variability from person to person because all those 3 people took the same dose, and one person was almost tripping, but the other people were like, I don't feel anything.
With like, seems like a lot of stuff, it's sort of like experiment on myself, like, try like 1 gram, like what do I feel?
That's probably
give it a few days, and then maybe try like a little less or a little more.
And people tried, like, sometimes people take very small amounts every day, or sometimes people will take like a gram once every 3 days. It kind of is whatever works for you.
Because I've heard some people say like you shouldn't take it every day.
I would probably go along that end unless you're trying to take like the dose that you don't feel anything and you're just trying to get like a little bit of something. But again, it's kind of what works for you, and everybody has such a different variability, especially on. I wish we all, when we say mushrooms, I wish we all meant the exact same thing. There's so many different species, so many different ways to get it, so it's like, it's so variability.
Well, it'd be nice, and I appreciate you sort of sharing what you did on that, because I think it at least gives me sort of an idea of where to start. I may end up, thinking like, I need to do less or more. That's just like that effects from the microdosing, but I've always been curious about that, because I've certainly like macrodosed and had good visuals, and that's great, but like the medical benefits, I'm very curious about that. So
definitely try. I would recommend checking out Arrowwood too, and you can read on their experience reports, so you can see what people were experiencing based on the doses. So you can look at light doses, see what worked for people as well, and see if that compares to your own.
EROWID.
Yes.
Yeah. And if you Google psilocybin mushroom dosing calculator,
that's awesome.
Amazing piece of software on it. Memory.org.
That's great.
You put in what species it is, whether it's dry or fresh, and what level of effect you want. And
there was a meetup a little while ago on elder psychonauts that was focused just on microdosing, and that was very helpful to have people share those experiences and the 3 days on or one, the regimen for on days and off days and the sizes of the dosing. And maybe be a good idea to kind of fish around to put together a group like that.
I was going to say, actually, that is one of the events that I had planned for August, and that'll happen on August 17th. It's not up yet, I do apologize, but it's focused around a discussion around microdosing. We had a picnic in Gabriel Park in Multnomah Village with the Psychedelic Society a few weeks ago. And that was one of the topics that kept coming up, is people sharing their individual experiences with microdosing and how much is working for you, what are the effects, blah, 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 what are you using it to treat, what are your intentions with it. I thought that was awesome. Thank you for that again. That'll be online.
Yes, I have two questions actually. What can you use to treat meth addiction?
So specifically, there are many different things, and methamphetamine is one of the hardest drugs to quit. In fact, there's only one other drug that's known to be harder to quit, and that's tobacco. Methamphetamine, basically, so we have, it's not the best pen. It's a little better. So that's my beautifully drawn synapse. So we have the axon terminal with the membrane vesicles in it, and then we have the receptors down here, and we might have a few. So, and there's a little reuptake transporter that is, so normally neurotransmitters will come down, bind to these receptors, and they will get sucked back up inside through here and either destroyed by the MAOs or repackaged back in the vesicles. So what cocaine does is cocaine blocks this receptor from pulling it back in. So you get more and more kind of building up because the machine that sucks it back up is not working. Methamphetamine's a little bit more sinister in its drug action. So methamphetamine actually works its way into here, empties all of these vesicles down, and has these transporters now start working in reverse. Wow. So it literally will flush out all your dopamine. So all this just comes out, and which is why it's so much more, it's neurotoxic, because if you get so much dopamine down there, on top of the, cocaine just blocks this versus methamphetamine reverses it. So, there are many different kind of things that people use. I kind of geared that ayahuasca treatment towards somebody with a methamphetamine thing because I was talking about dopamine so much, and that ayahuasca DMT seems to have a specific effect on dopamine modulation. So I could probably advocate that things like psychedelic use would be helpful, although there's some pretty basic things that are more in the legal realm. There's something called conditioned withdrawal. So that's kind of when you, even if somebody were to go to a rehab and then go back to their home, like maybe where they were using methamphetamine, just seeing that chair that they used to sit in and smoke actually activates the withdrawal in their brain, especially kind of the friend group, possibly if they're still associating themselves with things like that. So well, actually, a very interesting way to kind of combat drug addiction would be throw that chair away, get rid of that chair, rearrange your apartment. And I find kind of what I was mentioning in the psychological thing, social connection. So kind of building that social connection, building strong bonds with people, which why psychedelic use might be really helpful to kind of like, because they're connecting medicines.
Right.
If you have like a group of people, it can be maybe even if the actual psychedelic is providing some benefit physiologically, just that connection with other people, having that tight friend circle who are willing to be so close and open and share can be all the world. But again, it's a very difficult drug to overcome. And there's steps that we can take to help people who are suffering from that.
Cool. And then the other question was, with the 3 volunteers, why was the left side lit up?
So there was different regions of the brain, and delta waves tend to be, off the top of my head, kind of the longer waves. So everybody was kind of experiencing different cognitive effects. It's a little hard to say exactly why it was happening. Right now we're at the point of just kind of giving and looking and then figuring it out, but we're hoping that future researchers will be able to kind of do that direct thing. But the left side of the brain, you have different parts of the brain, like certain parts.
The left side is logic and the right side is creative, so I'm surprised it didn't stimulate the right side.
So it's actually really interesting. That is somewhat of a myth, that it actually different parts of the brain. So for example, we have amygdala, and the amygdala is on one half, it's on the other half. It's kind of 2 parts, but they're doing kind of different things at the same time. There was this really interesting experiment that was done where a researcher got somebody who had lesions in one half of the brain and the other half, and people who didn't. And this is poorly drawn. It's supposed to be kind of in an S shape. So a person with damage, I always get them mixed up, but I believe it's the left half, they could see only the S. They couldn't see the Fs. So it was the big picture idea. Versus somebody who had damage to the other side only saw the Fs, but they're like, what, there's an S there? I don't know what you're talking about. So it's kind of not so much creativity and logic, but big picture versus details.
Yeah.
So.
Okay, thank you.
Yeah, definitely.
First of all, thank you. This has been amazingly helpful. Also sort of exhausting. It's overwhelming.
Sorry, I know I was like throwing in all the information, which is why I'm offering these slides to anybody if they want to look at them and kind of break it down a little slower. And if you have any questions, you can shoot me an email. I'd be happy to talk about it further. Or if you have a question that's not necessarily related to this, I'd be happy to break it down.
So I had 150 questions. I forgot all of them. but towards the end, he said one thing that I still remember, which was advising against the illegal use of these things. And I'm in disagreement with you about that. So I'm wondering whether you could elaborate on that a little bit.
I mentioned that more in the clinical and research perspective side of things, because if like the clinicians are kind of doing that, I mean, there's kind of ways to get around, not around these laws, but for example, there's an ibogaine clinic in Mexico where people can travel to, or there's ayahuasca things in Peru. I'm not trying to say that we shouldn't use these medicines because they're absolutely amazing. I'm just saying if somebody is trying to advocate for policy differences, kind of violating these laws probably would not be that helpful. So I was specifically talking about the researcher or clinician aspect of things rather than the exact person just consuming it. And that's not to say that I'm obviously a proponent of doing all of these things. I just think that if they're done, they should be tried to be done in the legality of the law. There's also, for example, Oakland decriminalized all plant psychedelics. So they decriminalized magic mushrooms, psilocybin, they decriminalized DMT and ayahuasca, they decriminalized ibogaine, and they decriminalized peyote, mescaline.
So I went down there, I couldn't find a thing.
Oh, maybe it's a bring your own policy.
I was all over that town.
So when I'm talking about stay within the legality of the law, I'm not saying don't practice, but maybe open up a clinic in Oakland or something like that, or Denver, just decriminalize mushrooms. So things like, kind of like trying to stay with the policy as much as we can, but I'm not trying to say let's not use it.
Say again why you think that's a good thing?
Because if we are basically breaking the laws. It'll make the people who are in charge of the policymakers try to criminalize this entire industry and kind of everything that we're doing.
That's what happened in the 70s.
But then there's sort of this fine line, because technically this stuff is illegal, but then it's like, this was just decriminalized. It's still illegal there, but it's like a low priority. So it's like, you know, how do you approach this? It's sort of like, you want to research it, but it's like, you can't just totally like...
Carefully.
Yeah.
Absolutely.
Without there being any money for research, you know, we're talking 20 years for some of these things to get to be legal.
And that's
in the meantime, like with PTSD, we have, you know, millions of people who are suffering.
Absolutely.
I mean, it seems to me immoral to agree to
Make them suffer.
to hold off for 20 years. You know, people die during that time.
Absolutely. And I'm in complete agreement with you. For example, if a veteran, like cannabis has been shown to be helpful with PTSD, but if a veteran was to smoke cannabis, they risk losing their VA rights, which is absolutely atrocious. And I wouldn't want somebody to lose their VA rights. So I'm saying maybe we can find ways to kind of like, be careful about it, stay within the legality of the law, but still find ways to get that medicine. And things are changing. For example, MAPS is a 503c nonprofit, so you can donate money to them, and that will be a tax write-off. And then they fund these research programs. They're working with the FDA, and they've basically got what's called, I'm forgetting off the top of my head, but they're able, even though MDMA is Schedule 1 right now, they actually have the legality to practice it in clinics all around the United States.
And
so
the one here in Portland probably will open soon.
Exactly.
But that's one drug, and you have to have a PTSD diagnosis and have failed 2 medicines in order to get in, and then have, you know, probably thousands of dollars to pay for the medicines.
And we need to change that.
I mean, yes, it's a victory, but it's got a lot of asterisks along with it.
Absolutely, which is why we really need to kind of push this. And I think the best way to get to that would be through education and policy and making us seem that this is a complete legitimate science and we are not criminals in any sense of the word.
Yeah, well,
so
I just disagree with strategy.
Absolutely.
I think doing illegal actions and creating a culture where we're doing what we're supposed to be doing.
Absolutely, and I'm not advocating against that. No, I love that. I love that. Exactly. It's a good to play devil's advocate. And definitely, if somebody is experiencing PTSD and looks at the research and is realizing, hey, MDMA is going to work, go ahead, do what works for you. I'm a full...
I've worked with it. It's incredible.
Absolutely.
I mean, it's just incredible.
Absolutely.
And that is being held back from people is insane.
Yeah.
Absolutely.
Definitely lobby Big Pharma.
Could you talk about neuronal health and spacing between doses? I read about MDMA and then you need to wait 4 to 6 weeks, and also about macrodosing, psilocybin.
So everything needs to be respected and everything in moderation. Like I was mentioning earlier, you can die from drinking too much water. So it's really good to kind of let your body rest and relax, and bigger gaps are always healthier to kind of help like improve neuronal, like to kind of regenerate the neurons inside of your brain, to help up regulation, to help down regulation. Again, probably if it's something that needs to be consumed more regularly, if that's helpful, kind of smaller doses and things like that to kind of take into consideration. But it's definitely good to just kind of be mindful that these are very powerful substances that we need to respect. I hope that was a all right answer.
Thank you.
So back to the MDMA study, which is currently phase 3. What do you know about the timeline, since phase 2 was incredible and obviously shows that it's working? I think we have hundreds of veterans every day killing themselves because of PTSD, so we can't really wait that much longer. So
Absolutely.
What do you think is the timeline going to be?
It's hard to say, especially with our current political administrations that are around right now. Actually, the FDA we were supposed to have the MDMA clinic up in January here in Portland, but the FDA, due to our current administration, actually put a huge halt on that, especially with the whole government shutdown that occurred. So it's kind of hard to give you a solid answer. I'm hoping as soon as possible, but with the way things are currently going, it's really hard to say, especially with like if we have more government shutdowns, or the whole government's starting to get divided against itself. That's really not going to help things that's happening right now. And kind of bouncing off to what you were saying, I think if somebody is in need of treatment immediately, we should find ways to do it. For example, there are certain places that use religion as kind of a means to get the legality for that. So like that's what I'm saying, like maybe we could say within the law, establish like kind of spiritual places that we can apply these medicines to, and kind of stay within the legality of the law. So but again, it's kind of hard to give a total answer on that. I believe the Stage 2 trials took a little while, and hopefully it'll be done in the next few years. I know MAP's ultimate goal is to just make completely legal, basically, facilities where kind of anybody with anything can come in and just use psychedelics in a safe setting. Kind of like, not necessarily a cafe, but just a place where there would be like trip sitters available, people who are trained in psychotherapy and many other different things to kind of like watch over and guide through trips and stuff like that. But the ultimate goal is to make it accessible to everyone, even if you don't have a psychopathology.
Thank you so much.
Thank you.
Yes, thank you. Very helpful.